This technique uses instruments commonly found in most laboratory environments and allows for the analysis of several samples simultaneously. This method can provide key insights into how lipid metabolism enzymes are regulated in a cellular context and in different genetic backgrounds. Inoculate a colony from a yeast culture plate into 20 milliliters of SC media containing 2% dextrose and incubate at 30 degrees Celsius for overnight with shaking at 200 RPM. On the next day, dilute the culture in 50 milliliters of fresh media to a final OD of 0.2 and grow it for 24 hours until the stationary phase is reached. Prepare two 20 milliliter aliquots of quenching buffer for each sample and store them at -80 degrees Celsius. Simultaneously, prepare radiolabeling media by adding acetic acid sodium salt to dextrose-free SC media at a final concentration of 10 microcurie per milliliter. Collect the cells by centrifuging at 4,100 times G for 10 minutes. Remove the supernatant and wash the cell pellet once with 20 milliliters of dextrose-free SC media. Collect the cells again by centrifuging at 4,100 times G for 5 minutes and transfer them to a labeled 2 milliliter microcentrifuge tube using dextrose-free media. Centrifuge the cells for another 2 minutes. Resuspend the cells in 500 microliters of dextrose-free SC media and begin the radiolabeling period by quickly adding 500 microliters of radiolabeling media to each cell suspension. Incubate the tubes in a rotating incubator at 30 degrees Celsius.